A common refrain in the area of therapeutic bacteria is the problem of strong immune responses to the bacteria. Though there aren't many engineering studies involving the alteration of these properties, there are a few. In this study by Species and coworkers, the authors demonstrate that the outer membrane lipid can be altered to mimic the structure present in another bacterium, Chlamydia. If we examine the surface of a gram-negative bacterium, there is the cytoplasm, then the inner membrane, then the periplasm, then the outer membrane. The lipid component of the outer membrane is called lipid A and is composed of a core oligosaccharide connected to 6 acyl chains. In your laboratory strains of E. coli, this is the extent of the cell surface. There aren't additional modifications to the surface or to these lipids. In commensal and pathogenic strains of E. coli, there are additional carbohydrates called O-antigens that are covalently attached to the LPS. Additionally, there are often K-capsules that are attached by lipid linkers to the outer membrane. Though these capsular polysaccharides play major roles in adaptive immunity and the protection of the bacterium from complement, they aren't the feature of the bacterium that elicits strong innate immune responses. It is actually this lipid A moiety, the protein component of the outer membrane lipoprotein, LPP, and various other signals that elicit these responses. Collectively, they are referred to as PAMPs, or pathogen-associated molecular patterns. In other bacteria such as Chlamydia and Legionella, this core lipid A moiety has a different structure and has a very different effect on the innate immune response. They actually antagonize the response. During normal lipid A biosynthesis, a series of transferase reactions assembles the oligosaccharide and decorates it with specific acyl chains at specific positions. Different bacterial genuses produce different lipid A's, and the variation is in the identity and number of sugars, the presence of phosphates, and the length, position, and count of acyl chains that get attached. Chlamydia produces a 5-acyl chain variant. It is known that often the pentaacylated lipid A's are less stimulating of toll-like receptor 4 and thus a weaker immune response. If you mutate this last gene of the E. coli pathway, MSBB, you get an entirely viable bacterium that decreases interleukin-2 responses in mice twofold and is reported to have an even stronger effect on the human immune response. In this study, the authors swap out the first gene of the pathway for the cognate gene from Chlamydia. The resulting E. coli variants are viable and produce altered LPSs. But it results in a spectrum of products. They end up with at least 6 different variants of LPS that are hybrids of the hexa and pentaacylated variants.